So today's webinar is going to be on the GDC API and apps. My name is Himanshu Sani. I'm with Biomedical Research. I'll give you an overview on the API, some key features, and some references where we can get more information on. So regarding the API, so the GDC API provides developers with the programmatic interface to the query and download features of GDC, to the GDC data, metadata, annotations, and also to submit data to GDC via the API. It is the external-facing REST interface to GDC. It uses JSON as its communication format and standard HTTP methods like get, put, post, and delete. So delete input for the submission. Also, the GDC API itself supports the GDC data and submission portals, and I'll go through that a little bit more detail in the next couple of slides. And the GDC data transfer tool, DTT, also underlying uses the GDC API. So, and there's also a GraphQL interface separate to the REST API, which provides a little bit more flexibility. But I'm not going to cover that in today's webinar, but I will point to where you can get more information on that towards the end. So in regards to how the GDC uses the API, so here's a diagram of a GDC architecture, and you can see either through the web browser web portal you're coming through, GDC portal, submission portal, or the DTT. Once you get down to the legacy portal or any of the other interfaces of GDC, all the communication goes through the GDC APIs. And those APIs underlying work with the Elasticsearch, our Elasticsearch, and the data storage to bring back the results. Also, I just wanted to point out that the API was one of the core principles when we started GDC to be, so we designed it so that it is a core principle of GDC, that the API is a modular that underlying all the portals, all the external faces of GDC runs on a RESTful API so that it can be independently. This allows us to be more scalable and deploy the API separately. So this was one of the things that we wanted from start get-go to be part of GDC's architecture. So I'll go over the API key features now. First, some things I want to cover is the format and various search, download, BAM slicing, analysis, and submit features, and I'll go into a little bit more detail of them. So the first thing is the GDC API format. So all GDC, of course, the API works through an endpoint. In this case, an example is the project's endpoint. And then there are the query parameters, such as in this case, field and pretty. So those are the two parameters we're displaying right now, and I'll go into a little bit more details how the structure is created, how the queries run. And the return by default is JSON, but we support TSV also and XML in some cases. We'll come back to that in a second too. So the various endpoints besides projects I just showed is status, files, file versioning, case, which provides you the case metadata, history, annotations, and then mapping, which is kind of interesting. We'll get to that too. It's a very cool feature that gives you information about the metadata for creating the API. Metadata for the query parameters for the API. And the various query parameters, I've only listed some of the main ones. There are more, and you can get them from the user's guide, but the main ones are the facet, which lets you get aggregate data, filters, which lets you specify which you want to filter on some specific things. So if you want to say, give me a particular submitter ID or a particular file ID, you would use the filters query parameters. If you want to not bring back everything in relation to an endpoint, you can specify which fields to return, and those fields takes care of that. And then the format, return format as a JSON is default, but if you want to bring back TSV or XML, then you would tell it format equal to XML or TSV. Expand is also an interesting feature. I'll get into that a little bit more, and gives you a lot more flexibility and power of how to move, what you can return from the API. Size, we paginate through the results, so you don't get back if the results are more than 10, then you only get back 10 as a size as default, and you can change the size parameter to increase or decrease it as you choose. So same thing with size. There's a format from parameter which lets you paginate through it, and sorting, and then pretty. So these are just some of them we'll go over. So with that, I will go to some of the demo of the search features. And so the first thing I wanted to display demo was the status. By the way, I'm using an API tester. It's the Chrome built-in extension. It comes from Chrome. It is on our page to get to it. And I mean, if you just Google Chrome API tester, you'll get to it, and you can download it yourself. It's fairly, I use it fairly often to test or check some API functionality. So it's fairly easy to use. And I just created these upfront, and you can save them. Actually, I use it for the testing of our new features and stuff quite often using this tool. So in this case, I'm trying to go over the status. So just display status. And I'm going to close the header. So we show and then this case is showing me the release date, status, tag, version. And it kind of, if you go to the application, GDC data portal, you'll see at the bottom this information where the API version and then this is coming exactly from that status call that I just made. So I'll try to go back and forth, rotate between the data portal and the API to show how the actual GDC data portal uses the API underlying it, and then you can figure out similar queries using API. So let's go to the search functionality. So in this case, I want to bring back, so if I was doing a project search, bring back all projects that are currently housed in GDC, and through the data portal on the projects tab, it's giving me a listing of projects in GDC. Right now, I think we have like 63 projects supported by GDC, and then it's giving me different programs. How many projects within each program are supported by GDC? TCG has 33, Target has 9, Genie has 8. We just released Genie about a couple of weeks ago, or less than a week ago, and we're really excited about it. So what you would do that query is basically here's the format, and let me go back to where I was. Couple of screens. So as I went through the URL projects and then the fields, this is really where I am, projects, and then I'm adding parameters, my query parameters, so it's the same format. So when I run this, okay, I need to click it. So it's bringing me back the pagination, information, so it's bringing back the total is 63. I put pretty as true so I can actually go to the raw. It's sometimes easier to read. And it's showing me pagination, there are total 63. It's showing me 10 at a time, and it's showing me all the information about each of the projects. So it's in no particular order because I didn't specify any kind of order into it. So this is pretty much what the portal is doing here. And I wanted to show the facets into it. So right now it's just showing me all the projects, 10 at a time. Go back to the pretty, so it brought back 10 projects. And now if I do is, I added, actually one thing I did add was the facets. So when you see, what you see is when I added the program named facets, besides the pagination and the hits, which had all the 10 projects, first 10 projects, there's an aggregation, and that aggregation is based on that facet I just added, facets program name. So it's aggregating everything by program name, and it's showing me in the bucket that it's aggregating it by program name, and then showing me there are TCGA, there are 33, target 9, so same thing. So that's exactly how these facets work. So that query is mimicking how the program, this program facet was aggregated. So that shows. So even though I only got 10 upfront, but I got all the aggregation underneath it for all the projects, all 63 projects. Now, another thing I wanted to show is filters. So I wanted to bring back, if I go to data portal, I want to bring back all CP-TAC projects. It looks like I have 2 CP-TAC projects under CP-TAC program. If I click on that, it's bringing me back CP-TAC 2 and 3. Under the API portal, I'm going to try to mimic the same query, and I have this is my filter for bring back program names, value CP-TAC, and I'm going to bring back sort by project ID. So I want to sort by whatever comes back by project IDs. If I click on that, it's brought me back 2, and now I want to. So it brought me back CP-TAC 2. And CP-TAC 3, and it's showing me some more information about it. There's a primary site associated with it, or the primary sites, and then the PHS IDs, dbGaP access code for each of the 2 projects. It's giving me some more metadata associated with it. So that's just a quick example of how the filters works within the, we covered the facets, filters, and sorting within this. Moving forward to, let's do some, so if you go to the other things you can do is GDC is a repository. You want to bring back files or cases, search on those. So we'll try to do one of those queries here. So we'll go to the file search, and I want to bring back all files that are VCF, all the VCF files within GDC, and I want to only bring back just the file name, the data format, size for those files. I don't want to bring back all the information. So let's see. In the GDC portal, if I did the same query, scroll down to format VCF, I see there are 159,733 files that are VCF files in GDC from the portal. And if I do this same query here, I'm going to go to the raw because I already put pretty equal to true. And so it brought me back 10, and it's showing me the total are 159,733. That's what I saw, and it brings me back the first 10 files for them. So this is the exact same query we did. And then the other thing was, once I added the file name, file size, and file format, those are the 3 parameters it returned me. If I remove that parameter, so basically I don't have that in my query, and resend the query, you can see it brought me back same information, but the hits for each of them, I have a lot more information now. I have the data type, file name, all the other information, MD5 sum, etc. But this is the field lets me, tailor my return if I don't want all that information. So there's another way of returning searching, and that is by UUID. I don't want to say this, which is that I want to only bring back on a particular file information, all the metadata associated with the file based on UUID. In this case, I don't really need to create any kind of query parameters except put the UUID / as my after the endpoint, file / endpoint, and I just put pretty equal true just to format it. And if I return that, so it brought me back all the metadata associated with that file. And I'm going to try to get the UUID right here. And if I go to the data portal, I got to that file, and if I click on it, it's and basically what you see here in the file summary page is what is returned by the API. So that's how that's populated for that. So that's the one-to-one query for it. And there's let's see, I'll go into a little bit more detail for some of the other things as we go on. So the other thing GDC allows you to do, the API allows you to do, is get some versioning information for files. So you want to know the versioning is usually at the bottom of it, so file version. So in this case, there's one version of a file. In the API, if I want to mimic it, there is two endpoints, file/versions, and then the UUID. That's the format. And I chose this UUID because this UUID was replaced, this file was replaced by a later version. And as you can see, this is my input UUID. It should say this is my input ID. And then the latest ID is giving me the different one. It's the same file, except that the version was updated because of, for whatever reason, the submitters updated the file and they added, whatever, and the information changed, so the UUID changed, but we keep the provenance of the file, and that lets you know which is it. So when you download from GDC data portal, you always get the latest file. But if you have some reason, and also when you go to the DTT, when you download a file, it will always download the latest file, even if you have the older UUID, and it will let you know that it's been replaced by a later one. It will let you download the latest one. But in order, if you wanted to figure out the versioning, you can do that. Here's another endpoint that does something similar. Well, it also supports versioning, but something in a different way. And in this case, the history tells me that particular file, there are two versions, so it's just giving me some information about it. There's version 1 and 2, what was UUID first and last, and which data release this version, this file was associated with. So originally it was version 12, and now it's in version 14. So let's move on to the case search. Cases can be searched in JDC through the repository, cases, or under exploration, cases, clinicals. The API, it's the same API for both. In this case, I want to bring back some results for a case. I want to bring back cases, actually in a particular case. So I want to bring back for a submitter ID. I have a submitter ID. I want to bring back all the information about that case. So it's the TCGA, BA, AOEA case. So I'll click on that. And I can see that it brought me back one case and gave me the metadata for that case. So it gave me, so I will do, let me try to show you on the portal this case. So we have a quick search. This is actually an API for a quick search too. I'm not demoing that today, but it's in the user guide. So I want to bring back that case. And this is the case entity page, and for that case, it's giving me the case ID, the project associated with it, how many files associated with that case, what's the experiment strategy for the files, and then it's giving me, demographic, diagnosis, family history information, metadata, clinical information about the case. So let's try to see if we can do that. So when I ran this case, it brought me back all the, sorry, I'm going to go back to it for a second. So it also brought me back the biospecimen information. What are the samples associated with that case? What are the portions? Analytes, aliquots associated with each of the portions and samples. So, and that's what's giving me over here for that case. So it's giving me the analyte IDs, submitter IDs for those analytes, and then the GDC UUIDs for them, and then the aliquot, disease type, portion, etc. for them. So it's giving me all that information that you see on that page. And it's showing me that this is one count. It's brought me back. Now, I wanted to use the expand feature with that case. So the case we just saw is the same case, and I'm bringing back for a case. I want to bring back the expand feature. So I want to show what that means and what the expand gives me. So if I bring back this case, I want to make sure it's the same one. I believe it is, but let's make sure. So that's the same case I have on top, and I want to show what the expand. So this is the same query we just did, and I want to add another query parameter. Diagnosis. I want to expand the diagnosis. And what I see is that it added the diagnosis into my query. And I can expand on the diagnosis. It's telling me year of diagnosis, classification of tumor, which is not reported, primary diagnosis, tumor, all the pertinent information, ICD code. And that kind of is what was under the diagnosis tab. Sorry, I'll move a little bit slowly. So that's all the information you see under the diagnosis tab, which is coming from it expanded. So originally I didn't have that information here. Save it in this query, but when I added expand diagnosis, it added diagnosis. And you can do that for all the other things, all the other nodes that is supported off the case. So basically the query follows the GDC data model, and whatever is in the data model is how you expose it or expand on it. So the question is, how do we know what are the nodes that are supported by each of the files or cases or projects? And so let's do an endpoint on cases mapping. So that's the mapping lets you know how you can expand on it or what parameters you need to use for expanding on it. So I did mapping for cases, and it's telling me this is the aliquot IDs. It's telling me there's an annotations, and then there's annotations information for it. And I can scroll down, and it'll come down to demographic. It's giving me diagnosis we just went through. So if I wanted to just, so it's giving me all the different fields that comes underneath it. So there's 3 levels down. You can see cases, demographics, race, etc. So just want to go into raw and show you those a little bit easier to see all of them expanded. So if we went back to this one and we added expand into demographics, in this case we did a query. So now you'll see basically I changed it. I mean, you can add both of them. I expanded both in the, scroll down, it's further down, demographic information for that. So you can do diagnosis comma, diagnosis, etc. So that's another way of really expanding the API. And similarly, there's another endpoint for annotations, and I want to bring back all the annotations for a particular case. That's my use case. I want to have a submitter case ID, case submitter ID. I want to bring back all the annotations associated with it. So I created my filter in this case, and I'm doing pretty equal to true just to format it. And I want to bring back information for that. So it's giving me back, there are about all the annotations associated with it. So it's giving me the annotations various. So there's a no matching normal for this sample. Also, there are a few annotations associated with the analyte, same thing with the analyte slide, etc., because the sample didn't have any matching normal for it. So those are the various annotations, and there are 6 of them. So let's go up to that. So we will do in the case of GDC, I want to go into repository, and I want to bring back that case. And I want to bring back the annotations associated with it, so I can get to the annotations. I moved down one quick down, so actually I need to go to the other page. Sorry about that. That's the one I was trying to do. So browse annotations for that case. And that brings me back the annotations associated with that case. So going forward, let me go back to the presentation. So I want to go over the download, and the download API supports the data endpoint and the manifest endpoint. And the data allows you to download data based on UUIDs, and then the manifest will let you create a manifest for those UUIDs. And that's what the DDT data transfer tool uses. If the file you're trying to download is a control access file, then you need to pass in your token. If it is open access, then you're not required to pass in a token for it. Also, there is a related files, which brings you back all the other files associated with it, such as if you're downloading a BAM file and you want to also bring back the BAI file for it, so you would do a related file equal to true, brings you back. And then there's also a tar file, which will zip up all the files into a single tar. So let's, I'm not going to do a live demo on that, some of them because they're control access files and stuff. So, and what I decided to do was I ran them earlier when I created this presentation last Friday or Thursday, and then I basically cut and pasted from my terminal into here. So I just figured we can go over the format. So in this case, I'm just using curl, and I'm passing the remote name, remote header, and then my URL, and then I'm passing my UUID. So it's data UUID, and it downloads the file, and then it has gzip. If I have multiple file UUIDs, in this case, it actually creates a download tar zip for me with the date stamp, and it groups the two within it. If I have a control access file, then I need to save my token somewhere and then pass within my curl as a header the XAuth token, and then pass my token as a parameter and then download it. So that's the way a control access file works. And then if I wanted to create a manifest, the curl lets you do is a manifest, and then the UUIDs, I can pass in more than one UUID to it, and it will create me a manifest file for that. And the syntax of that is GDC manifest instead of download, and then gives me the date timestamp for it. That's kind of how the download API works. There's nothing more to it than this for the download API. So let's go to the next one, which is BAM slicing API. So GDC supports the data portal, and the API support BAM slicing. And this BAM slicing kind of works very similar to, the API works very similar to how SAM's tool or other such tools work, and has an endpoint called slicing view, and you pass in your query parameters for it, and it returns you the BAM file with the BAM header within that, the BAM header for that file within the BAM slides file, and some annotations with it. So the two parameters you can put in a BAM slicing is region, and you can do a chromosome, position, etc. within the BAM slicing region that you want to slice it by, or you can also do it by gen code, where you can pass in a gene, AGNC gene ID, such as TP53, GFR, etc. You can just do gen code equal to, and then your genes that you're interested in. The example I have is what the application does, similar to. So within each of the BAM file summary pages, you can do BAM slicing, and then within the BAM slicing, the example comes up is, this is what the drop down comes up, and you can say is, you can type in what are your regions you want to. We also started supporting recently unmapped, and then you would just type unmapped here instead of a chromosome position, and you'll get all the unmapped regions. So in this case, I'm trying to bring back chromosome 1, entire chromosome within that BAM, chromosome 2, 1,000 and more, and then chromosome 3, 1,000 to 2,000, and that's how basically, it's bringing me at that position at 1,000 and then 1 to 2,000, so you're bringing back that region. So it's the underlying, the application creates the exact same API underlying it and brings me back, and in this case, I could put in an output of get regions, brings me back the BAM file. So it's pretty straightforward, and the example I took out of it is from the user's guide. You can use it on the portal, and you can use it from the user's guide and test it with your. So it does require a token, since I am doing a BAM file, and BAM files are control data. So that's one other thing you need is both on the token you need to be logged in and have access to that file, authorization to that file, and same is required on the API between the two. And I'll go through some data analysis. So the another feature I wanted to show the GDC API supports is the data analysis, which is the GDC exploration page. And there are various endpoints. I didn't list all the endpoints here. I listed some of the high-level ones. There are a lot more of these, but I listed some of the high-level ones here, such as genes, mutations, SNVs, and then if you wanted to bring back for your cohort that you've done, all the top cases by gene count, all the top mutations, muted genes by project, and then muted cases by gene, and then muted cases by counter project, and then the KM plots. So they're all supported here. Let's do an example of them. See what's next. Yeah, demo. So let's demo one of them. So in this case, I want to bring back the metadata associated with the gene. In this case, I chose TP53, and I need the ID for that. TP53. Let's mimic that. So basically, I'm going to exploration, and I am bringing me back all genes, and I'm taking the first one, TP53, and I want to bring back information on TP53 as it relates to GDC exploration. And I found within GDC exploration, this is the gene symbol TP53, and then I have is the name, the synonyms for it, type, etc. And then it's giving me just a distribution of that gene within the various projects, TCGA projects, the SNVs for it, and over here, and then it's giving me the ratios within each of the projects. So if I do, so I do need, what does it accept is an ensemble ID for that gene. So I added that here, and it brought me back, that's the same query. So it's brought me back, the various information. The gene is equal to true. It's basically the same metadata, that description I just went through for that gene. That's the same information. So there are multiple API calls over here to create this page. So what I just went over is this metadata up front. But each of them are the API support separately for this page. So that's why it looks a little bit more terse here. And I want to, let's say, I want to bring back all the SSM occurrence for a case. So I have a case ID, so TCG case ID, and I want to bring back all the SSM occurrence for that case. So in this case, I'll try to do the same case, bring back results for it. So if I go here, go back to my exploration, I'll type in the case ID. And it brought back the case, and I can see the genes. So if I go to that case, let's see. Sorry, let me go back here, and I want to clear this. So it brought me back and said there were about 30 mutations for it, because those were just the for that case, and it's bringing back all 30 mutations for that case. If I go to the case and I click on that case entity page, at the bottom of it, also it'll give me information on the mutations. So the most frequently somatic mutations for that case is showing me those are 30 mutations for that case associated with it, and it's paginating 10 at a time, 20 at a time. So it's updating it and giving me all the metadata associated with that, consequences and information for that case. So now I'm going to display the same thing. So in this case, what I did was I did the size 50. So I want to bring back all the mutations at the same time for that case. So if I send it there, create it, and I go to the, like maybe pretty easier this way. So if I do so it's showing me their 30 return, so which is the same thing we got on the over here, and it's showing me all the information. And because I did size 50, so it brought back all 30, and for me total was 30, so it brought me back all 30 over here. And I can expand on each one, and it's giving me the ID for it. It's giving me the chromosome number for it and ID, and I'll go on it a little bit more. So as you saw on this page, I had more than just the ID. I had the consequences, I had the impact. So how do we get that besides the ID? So let's do the same query, and in this case, I'll do is expand on the consequence transcript. So the same query, I'm using the expand feature again, and in this case, you'll see what it did was it brought back the so if I click on the consequences, it expanded it and gives me on the transcript, and it's giving me a lot more information for it. It's giving me the transcript ID, the AA change, the reference sequence, refseq ID for it. So basically, that's how you would do. So each one of them is, you can expand on it and get more information for them. I think that kind of covers my, just a little synapse of how the API works, the analysis API works. Now let's go to, so another feature that GDC API covers is the submission. And submission is, you need to have a submitter role, not just a data downloader role for GDC. So within the people who are designated as submitters for a project can use the GDC submission portal to do the submission, and they can also use the API. So the syntax kind of goes for the endpoint, says submission, your program dot name, project code dot code, and for example, TCG ALCH would be an example, And when you log into the submission portal, you will see that on top, the program name and your project name, how GDC assigns it. It's a secure way of submitting also the experimental data files. So you need a token to submit your data files through the API. Also, you can update and create. So if you want to delete files, you have to use the API for it, something that you put in earlier and was a mistake and you wanted to correct something, the API does it, allows you to do it, or you can go to user support, which will help you do it too for it. So syntax kind of works is you create a request, a JSON request. In this case, I'm creating a project ID. That's my project ID. I'm trying to submit a case. I want to create a case. And this is the same thing functionality supported by the GDC submission portal. So you can do it as CSV or JSON with the submission portal. This is the exact same thing, but you're doing it here through the API, and I'm putting a submitter ID for it, and I'm associating the project with it. And then basically I'm passing my token. I have a token, and I'm creating a curl post, and I'm passing in my request, this request, as a data binary, and I'm passing in my JSON, because as a JSON time I'm passing, and this is my URL for it. And I want to do it as asynchronous, so I can submit it as asynchronously equal to true. And then once it is accepted, you will get back a code. If it is valid, you will get back a transaction completed and a transaction ID. If there are issues with your submission, then you will get back response to error codes and error message within that. That's kind of how the submission API works. There are more examples in the user's guide for it. Another endpoint that GDC supports is the GDC legacy archive. These are files, the unharmonized or AG38 files that GDC, when it started, that previous GDC that came from CG Hub, and those are maintained within the GDC legacy archive. Basically here, and the difference is that when we are supporting them, you need to add legacy endpoint and then basically wherever the search endpoint is. So in this case, I'm doing it by cases. I want to bring back a particular case. All the primary site is blood. All the blood cases I want to bring back in GDC. So I want to do that using the API. So it works exactly the same as what we went through the API cases files before the search API, except I have a legacy added to it, annotated to the qual. So one I just showed you, I want to just run it, and so it brought me back, all the cases, all the total files, which are, in this case, there are about, it's telling me there are 2,862 files that fit my query parameter, and it's bringing me back 10 at a time as pagination, and I can do a to and from query parameters to paginate through them. So let's see. So that pretty much covers the key features of GDC API. I want to give you some references and user guide links, which are kind of useful. The first one is the GDC Users Guide. I already opened it, clicked on it before, and that's in the GDC Documentation Users Guide. And this has a lot of all the features that I went through: search, retrieval, download, analysis, advanced slicing, submission, and it also has lots of examples: Python examples, GraphQL examples, curl examples. There's lots of, and then how to use the fields, etc. So I would really highly recommend to go through it and use it. User support team has done a great job of documenting the API with lots of examples here. Another thing I want to cover is there's a GDC Community Tools page. You can, within the API, within this presentation, this is the link for it, Community Access. So it's on the GDC website, Community Tools page. And the Community Tools page lists all the tools that either the GDC User Services team on their own have created based on user requests. We get a lot of requests, and and based on them, sometimes they try to codify that into a tool and put it on the external on this page. So for example, GDC read groups page, Jeremiah created that, and then there's various other ones also that other entities outside of GDC have created tools, and they have provided us access to them. So we try to list them on this page. The tools are maintained by the authors themselves, not GDC, even though this is, this may be created by part of the GDC user support team, but the author is the one that's maintaining that page. So just keep in mind for that. There's the Git repository and then the repository for it. So, and then same thing you can do for the various other tools that created. And I think this is, for me, a really useful page because it shows how people have used the GDC API. So all these tools have been built on the GDC API I just went through for the various ones. It shows there's one I wanted to show was the R package. Yeah, so there's Bioconductor has created various tools. I think Sean Davis created these. He moved from TCGA, used to work on TCGA into GDC, uses GDC API, which is an R pack, Bioconductor package for it, which is kind of useful. And then the GDC repo repository also. Not everything is open in GDC currently, but whatever is open source you can see that here, and you can get to that within GDC. And then I have various references that we have in GDC, the data portal, submission portal, the things that I just touched through. I think that's the end of my presentation, Sharon. If there are questions, I'll be happy to answer if I can.
Yes, Himansa, there's several questions. The first question, is it possible to search for particular variants in VCF files, say those that overlap exons of BRCA1?
Say it again, Sharon, sorry.
Is it possible to search for particular variants in VCF files, say those that overlap exons of BRCA1?
I don't think we're downloading the VCF. We don't have a search within the VCF files. Bill, if you can answer that, I haven't tested that, or I don't believe we support within the VCF files search through. But you can get to the VCF files. Bill, can you answer that, or is he not able to?
Hi, this is Bill. So within the GDC, there is, this can be queried using the SSM's endpoint. This does not come directly from the VCS files. Rather, it comes from a somatic variant filtered version of the VCF files that were converted to a MAF. And this is what can be queried in the exploration page on the GDC portal. So it's not quite the VCF, but it is kind of a sanitized and filtered down version that should only contain somatic mutations.
So I would say the examples I showed, the SSM over here, those are the ones, basically we took the VCFs and created the map files out of them, and that's what indexed, and that's what you can search on, right? So.
Okay.
So I would give this a try. Go ahead.
The next question, if for the sake of consistency in a project, we want to query always the same release, not necessarily the last one, how do we do that?
So, as in the sense of release of files or data, or?
Yeah. I mean, if they want to query for a particular release, like release, or data release 15, how would they do that?
So I don't believe we support that. We support the latest release for the data. So we annotate it, but the only way if you're interested in the files, you can go back to the files provenance and then find out which release associated with it. But I don't believe we would support each version of the previous releases. It's just too much data we have in GDC already that we're supporting. Bill, you want to answer that? Is that correct?
Yes, I can add to that. So while the version can be queried, the version doesn't always correspond to the exact release. For example, if I had 100 files in release 1, those would be version 1. Now I submit 200 files, 100 of which overlap and are new versions of the first 100. You would have 100 files of version 2 and 100 files of version 1, all from the same release. So querying the release number wouldn't be a good way to do that. Now this doesn't have much to do with the API, but for that use case, one could go to the GDC documentation website under the Data tab, under the Release Notes link. And the Data Release Notes actually for each release has a manifest. So one can filter down by project and data type, and you could download all of a certain type from a certain project, all from the exact same release.
So, so you're saying is go to the data portal and then go to the release notes, and then each of the release notes associated with it. So if I'm interested in release 1.16, and if I scroll down, I can get to the links for the...
Sorry, go to the data.
Oh, sorry, wrong one, right. Data, release notes, data, and if I do 1.6, those are the manifests. So those are all the files associated with that particular release, and that's how you can get to it easily.
Okay. Are there notes available for later study? We are recording this presentation, and that will be made available on the GDC website. The demo that Himansu's done using the Talent API, we do have an export of the JSON that's used for that. It's a JSON script we can also available on the website as well. Okay, the next one. On the GUI, there are some buttons that read JSON. How does this relate to the API?
Can you repeat that, Sharon?
On the GUI, there are some buttons that read JSON. How does this relate to the API?
On the GUI, some buttons that read JSON, you said?
Yeah, well, the question is that read JSON, but I think, you know how you can download files in JSON format, like clinical biospecimen. How does that relate to the API?
Hey, this is Bill. I can answer that.
Okay.
So when you go on the GDC portal and you're filtering down files or you're looking in your cart, there's usually a button there that says JSON or export, and then you can select JSON. This relates to the API in that it's kind of sometimes it can be a specific query that will convert to JSON, a set of pre-selected fields that are normally useful for the particular set of files you're looking at. Other times, it's just kind of a JSON export of whatever you're looking at the screen. So since everything on the GDC portal uses the API, it just kind of runs through and gives the same query that the portal is doing directly to the user.
So basically, GDC supports JSON as the format, both for input of the query and also the response. And you can change the format of the response to XML or TSV, but default is JSON. I don't know if that answered the question. Go ahead, Sharon.
Okay, does size have an all value so that all hits can be retrieved?
No, it doesn't. So the way you would need to do that is you need to come to the pagination and you need to figure out what is the total size and do it. There is no all. Am I right, Bill?
That is correct. And normally what I like to do is just put a very large number that will definitely encompass my entire query. But the only thing I should warn you about that is if you put in an extremely large number, the API query will fail. So try to find a reasonably large number that isn't too large.
Yeah, and I was going to say so we do throttle the query. So if it is an IP that's using a lot of it, the bandwidth, we do try to notify the user if we know it, and we have had to block people because they're using cloud or something with hundreds of VMs and try to it affects our performance. So we do as good stewards of API users. I use a lot of APIs. I try to manage it so that I don't get blocked and try to use it. So I would suggest everyone to be good stewards of it and use the pagination as judiciously as you can, what makes sense for you. It does work. If you do put in a very large number, then it does take, as Bill said, very large time to bring back results. For me, it's better to throttle through a smaller number and get, and I can do next. It works a lot faster for me and a lot better for me to manage.
Okay, another question. Is there a way to query or call multiple or all UUIDs under a given project?
Under all UUIDs under a given project, yes. I believe you can do that if you do a files query and you want to bring back and give the project ID as your filter. That would do that. The other way is also this would be simulating the same thing as when you're going to the projects. I believe that's the way to do it, is filter it by the UUI, in your filter for files, do a project ID, and that will bring back all the files associated with it. Do you agree on that, Bill? That's the easiest way, I think.
Yes, I'd agree. Doing that from the projects page or from the repository page, just filtering by a particular project will give you all the UUIDs for all of the files within that project.
So, and you can do that under the API too. So files, and then you filter by the project ID. So you would do filter over here. So if I did.
Right. That was the last question, and we're just about out of time. Thanks everybody for joining today's webinar. Our next webinar will be January 27th from 2 o'clock to 3 o'clock p.m. Eastern. So please mark that on your calendar. We will be putting out notifications for this, and we will be making the recording of this video available on our GDC website, and we will tweet when that is available. Many thanks for your time. You all have a great rest of the day and a wonderful holiday. Thank you.